All right, come on in and have a seat. We are going to get started here in a second. I'd like to welcome to the stage Dr. Rebecca Reba-Wollman. She's going to give you an update on trial GSD 1A.
Perfect. Hi, everybody. Thank you for having me and welcome to our second day. I hope everybody is still wide and awake and there's coffee on the side. My name is Rebecca Reba-Wollman. I'm an endocrinologist in Connecticut. I'm going to be talking about Beam 301, which is a Beam Therapeutics trial in humans, focusing on base editing in GSD 1A. So I do serve as an investigator in clinical trials sponsored by Ultragenyx, as well as Beam Therapeutics. There are a few slides that are highlighted that are provided by Beam Therapeutics. Separately, again, I'm presenting this on behalf of myself, not the company, and I have additional slides based on published literature. So I'm going to say right up in front that really this talk is focused on it's focused on the science behind the human trial for Beam 301. I'm going to talk about the design of the human trial for Beam 301. And I will say at this point, I do not have human data yet for Beam 301, given the stage we are in the study. So again, at the end, I'm happy to take questions and I will answer everything to my best ability, but just in case somebody was anticipating that. Okay, so what are my objectives? I'm hoping to discuss what base editing does within the body and compare the biological basis of ongoing trials for gene therapy and nucleotide editing therapy. I'll explain kind of the nucleotide editing therapy. I'd like to differentiate an investigational therapy from an approved drug. So I'm only talking about an investigational therapy right now, so it is under study. This is not an approved drug that is currently used. We're going to talk about preclinical research. So that generally means mouse models and cell models of nucleotide editing therapy that then led to human trials. And then I'm going to provide some updates around phase 1-2 of Beam Therapeutics. Okay, so base editing. I've just used a number of different terms all together: gene editing, base editing, nucleotide editing. They're all upon the basis of something called gene editing, which is quite new. We're going to first say, what is a gene? So our DNA in our bodies carries our genetic information. That's what our development and function of all the cells and of our entire body is based upon. Okay, so we see this very long strand of DNA. DNA is wound up into these structures called chromosomes. That's how we carry it around so that they're small enough to fit in the inner part of our cell called the nucleus. And then a gene is a single stretch. So it's a segment of DNA that encodes the information for making proteins. Again, we have this massive strand, and so there are many genes on each one of these strands. Okay. So a gene is part of DNA, as I highlighted, is used as a template. So I don't think it allows me to do a pointer here. So you can see in the first image, I'm showing you a gene that is that segment of DNA. Your body uses that as a template to form RNA. And then ultimately, your body uses the RNA as a template to form a protein. Okay? If we use GSD type 1A as an example, the gene is G6PC. Again, that would be changed into an RNA, which would then be changed into a protein called glucose-6-phosphatase. Right now, so this study is only focusing on a more common mutation causing GSD1A. Over 30% of individuals with GSD1A, this is the mutation that they have, and specifically it's a point mutation. So I'm going to show you on some subsequent slides that it's hard to see here, but each one of these connections is effectively formed by a letter. That's a nucleotide. When one of those single letters is abnormal, that's called a point mutation. So the technique that we're talking about today does not fix a gene as a whole, it's specifically targeting to this specific point mutation. Not all mutations are point mutations, and changing it back, okay? Ultimately, a lot of people know the term R83C. Again, that is the abnormality in the protein that causes glycogen storage. That is one of the causes of glycogen storage disease type 1A. So I'm going to acknowledge, I've given this talk. Dr. Diva De Leon gave a talk on ultragenics. We've done it on prior studies, so a lot of people are familiar. Again, my talk is not focusing on the ultragenics pharmaceutical study, but I'm going to acknowledge that some of this, I think, helps once you've understood one, it's easier to understand another. I'm going to acknowledge there was an RNA trial focused on GSD1A that is unfortunately not currently ongoing. But to focus on briefly some of the differences, so whenever you have a genetic type therapy, you need to carry it in something. You need to carry it in a bubble to get it to where you want to go. The ultragenics product is something called a non-integrating gene therapy. So it means it drops off effectively that little stretch of gene, in addition to some other things that help that gene work, drops that off into the cell where it kind of lives independently of your own DNA. The bubble is an adenoviral-associated vector, or an AAV8. So there were some talks talking about AAVs as viral vectors, and again, that's the carrier. And that trial has completed a phase 1/2, is on to a phase 3. So what that means is, in general, phase 1/2 is initially, this is first in humans. So it's focused on the safety of the potential product in humans. It's focusing on finding the correct dose for that drug, and then ultimately it's a small trial because of that. And so most classically, and there's going to be a talk later on today about some clinical trials, but because it can be a little different in rare disease, but most classically you then move into a phase 3, which is a much bigger study. Again, with the ultragenic study, it was approximately 50 people, and that's really so that you can have two groups to really compare how much of a benefit this is versus the risks. So I explain all that because the ultragenics trial is completed phase 3, has long-term monitoring, as you heard about yesterday, and was announced at the start of this that, again, they are waiting to see results of an FDA review. So some of the differences briefly with nucleotide editing is, first of all, that carrier bubble. So the carrier bubble, and I'm going to comment a little bit on the safety on the next slide, it's called a lipid nanoparticle, or an LNP. And so instead of a virus, and again, it's a virus that kind of all the viral elements that can make you feel sick generally are largely removed, versus a lipid nanoparticle is really kind of a bubble made out of fat, truly. And I'd say we benefit as a group of individuals who think about glycogen storage disease, that many of the glycogen storage disease are due to the liver disease, and the liver is very good at picking up fats. And so this is a lipid nanoparticle. And then the inside, I'm going to go into a little bit more. It's multiple stretches of RNA that I'll give some more description. One of the biggest differences, though, is the study I'm talking about is a phase 1/2, so not at that bigger study, bigger efficacy assessment step. So I'm going to say briefly, again, so whenever I think of a study, and I tried to organize this how I would discuss it with a patient, to think about kind of the safety versus potential benefits in a study that we don't know about in humans, is what are some of the things that I consider. So first of all, to talk about that bubble. New is not better or worse, it's just we know less about it. So lipid nanoparticles are also a newer technique along with nucleotide editing. And so, but we use them in other drugs. Other medications that use lipid nanoparticles have risks of infusion-related inflammation and allergic reactions. So I tell everybody, for every drug discussion I have, any drug, somebody out there can be allergic, so we know that. But we know that some of these medications that often get lumped into a term called biologics do have some added risks for allergies and for inflammatory reaction, because the body really will recognize it differently than they'd recognize a Tylenol or something like that. We don't know what the risk is in this study, because we know that risk of the lipid nanoparticle changes. So even though these bubbles together, you can think about them having a similar risk profile, the percentage for each different type of this varies. But I'd say in approximately 20% that you can see reactions like inflammation, allergic reaction. I'd say most of those studies were done on the lipid nanoparticle COVID-19 vaccines that came from Moderna and Pfizer. And so those were injected under the skin, versus the product that I'm talking about is injected through a vein. I will say in those studies, that is the biggest number, that true severe allergic reactions, so it was more common to have mild allergic reaction, but a severe allergic reaction called anaphylaxis occurred in 21 patients out of about 2 million, so that is a very low rate. 7 of those individuals actually had a history of anaphylaxis, so may have some underlying risk to it. Okay, I'm really not going to go into the lipid nanoparticle further. So let's talk about, and this is one of the slides provided to me, what Beam 301 is. So it is an investigational, again, this is not an approved drug, one-time therapy that uses base editing to correct specifically the R83 mutation. That is one of the mutations that causes GSD1A. And I cannot show you on pointer on the left side of my screen. Again, you see those 2 letters. Those are nucleotides on each of the 2 strands of DNA that connect together. And again, there's an error in one that effectively causes an error on its mirror side. What Beam 301 contains in that lipid nanoparticle is, I describe it as 3 RNAs that build a machine and a matching strip. So the matching strip is called a guide RNA. So we're going to talk about what are the theoretical risks of a treatment like this, that this is going to edit somebody's own DNA. So you need to get the spot right. You want to line it up beyond as perfectly as possible because you don't want to change a different part of somebody's DNA. So that is the job of the guide RNA, to line it up. The other 2 RNAs essentially build a machine. Again, a lot of people have or should have heard about CRISPR. CRISPR technology won the Nobel Prize in 2020. And so this is a generation of CRISPR technology where CRISPR generally, classically, we think of it as inserting an entire gene into a mouse model or into a human. This is a modified CRISPR protein that's called an ABE, adenine base editor. So really it is built specifically, not just for GSD1A, it's specifically built for mutations that you just want to change that A letter that we see there. Okay, so this comes into the body and lines up and then uses these 2 proteins to fix one of the letters. And then the body actually comes in, it's only cutting one of the 2 strands of DNA that may have some safety benefits than cutting both strands. And then your body's own mechanism to kind of fix DNA, we have a lot of other proteins that's their job entirely, kind of comes, zips those 2 strands together and fixes the mirror on the other side. So I'm going to talk about this more, but that is kind of how it works. And I largely talked about this, right? So if this was in humans, you kind of wrap this little RNA machine into a lipid nanoparticle. You put it in through the IV. That is largely anticipated to go to the liver, even just based on that lipid bubble, but there's more specificity that I'll show. And then ultimately, it's going to target DNA editing. And with the anticipation, if we go back to that first image, right, you have a gene that has an error, which then you have an RNA that has an error, and then you have a protein that has an error. And it's that protein that doesn't work that causes glycogen storage disease type 1A. So the anticipation is if you fix the original gene, then you get a RNA template that is then corrected, and then you get a protein that is then corrected. Okay. So preclinical largely means animal and cell-based data, and this was published in Nature Communications in 2024. And so this is the primary science-based article that the human trial is based on. Okay, so I am going to go into some deeper science basis to this, but my hope and goal is to make some of this kind of more digestible. Okay. So as part of this, you actually have to create a mouse that has this specific mutation, has R83C. And so specifically, this study again, based on other prior studies, created a mouse that carries that specific mutation. And so you can see, again, this is a control mouse, generally a relative that does not have that mutation, versus this is the mouse. In their paper, they refer to it as HOM, as well as HU-R83C, means humanized R83C that was put into this mouse. You can see that they are much smaller. They largely kind of show the phenotype of individuals with GSD1A. Their liver weight is quite large compared to that control animal. Again, that's the comparison of a sibling who does not carry the mutation. Very low glucose 6-phosphatase activity. Again, we don't routinely anymore do liver biopsies, but for those of you, some of you, if you have GSD1A or another GSD, may have had a liver biopsy, so we know about activity from those old human samples as well as from animal models. So very low activity in this model of the mutation. And in fact, I'm going to say the number is 2.2 units, and so we know at this level, and I'm going to come back to it on another slide, really there is virtually no glucose 6-phosphatase activity. In addition, much like human individuals who have GSD1A, we saw much lower glucose, much higher triglycerides, much higher cholesterol, and an elevation in lactate. And I will say lactate response is a little different in mice than in humans. And I'm going to highlight that a few times, right? Humans are not mice, and so we learn a lot from these studies, but we have a lot more to learn, thus the phase 1/2 trial. Finally, again, uric acid was higher as well. So as I mentioned, some of it, that we see that reduced body weight, we see larger livers, severe fasting hypoglycemia. I'm also going to say some of the studies needed to be modified, and this has been seen in other GSD1A mouse models, that they do not survive very long, and so sometimes you have to use different techniques to be able to better assess them as older mice. Okay, so there have been prior studies looking at use of similar technology, CRISPR-Cas9, in mouse models to look at treating GSD1A. So there were 2 different techniques used. I won't go into it, but again, this is not the first trial. It's really incredible, all the dedicated people and laboratory-based research who make all of this happen to come to humans. I mentioned already that humanized mouse model. And I'll just say briefly, I'm going to mention it 2 times. Again, right, when you have a mouse, you want to try and think about everything that might be potentially helpful to learn about for in the future for human trials. That mice were treated at 2 time points, one as newborns and one at 3 weeks. Again, mice mature into adults very quickly compared to humans. And I won't get into it, but again, by doing this, they learned more about what doses were more effective in mice, what timing was more effective, and so that was done. So a lot of the studies are repeated on purpose within this paper to look at those different groups. Okay, so don't get lost in the figures. I'd say it's the next 3 slides that have more figures that really kind of at the top I try and put it into kind of more generalized terms because I can get lost in the figures as well. So as I mentioned, the first thing somebody would have to show to me to consider using this as humans is how specific is it? I mentioned that guide RNA. You do not want to edit other parts of your DNA. You want to be as specific as possible. So they needed to demonstrate this in animal models first. And so what they demonstrated on the left side of the slide is increasing doses, improved editing. I keep showing it like you can see it. So if you go from left to right on the different stage that's labeled B, as they went up on higher and higher doses, and I just put at the bottom 301L and 301H, those are the 2 dosing groups largely that they used in the animal models, that we can see as the dose got higher, the editing that when they looked at the tissue afterwards got higher, but the concern would be as you have higher and higher doses, how high is the risk of it editing the wrong area? So those are the gray bars, which again, you see increasing, but remaining relatively low compared to the main. There is always going to be some off-site editing. Biology is not perfect, but it is reassuring that it's at a very low level despite being at high doses. And so increasing dose increases the editing with a very low level of editing to other parts. And I'll say some of the terminology, slide B is looking within the same gene. So that's looking within that G6PC gene, right? It's very easy. You can think about it as slippery surface. If you have an RNA that's trying to line up, that guide RNA, it's very easy to shift a little bit within that same region. And so that first table is looking at a shift. Is it editing another part of that same gene? And it does. That's the low level that you see. But thankfully, that second edit doesn't cause a new disease. It causes GSD1A in a similar phenotype. And that it's a low level, right? If that was a very high level and you're like doing that same edit as frequently as you're doing the correction edit, it's not going to help, but that's a low level is reassuring. You also have the risk of affecting other genes in the body, and that's what really, again, as a physician, we need to be very aware of. We do not want to do harm while we're trying to treat an initial disease, and so that's what it needed to see. Number one, some of the studies are, where else is this lipid nanoparticle going? And I'll share again, we know lipid nanoparticles are actually not great at getting other places than the liver, and they do not see significant effects in other organs. But even more so, what about other genes? And so that needed to be assessed. And what ultimately, this later part, the uncorrected synonymous and the indels, are mutations in other part of the DNA, and that they did identify a second site, so a single site within a non-coding intron. So your genes have parts that create those RNA templates, and they have parts that do not. And so this was in one of those in-between parts, still can have effect on proteins and other parts of your body, but thankfully in that part, it is less likely to have an effect. And then they went on and used human models and actually computer models, which is used pretty routinely in medical science and human science as well, that demonstrated that this area was still a low-risk area that they do not anticipate would cause a new disease. Okay, so this is a busy slide. What I'd say is this is two different ways to show the same thing, and I just like the visible reproduction of this. So BEAM 301 demonstrates recovery of hepatic, that means liver, glucose 6-phosphatase activity. And so what I'd like to show you is, oh goodness, I really wish I had a pointer. What I'd like to show you is in figure A, right? So the white, let me see. The white and the gray are serving as controls. I'll say that these are similar aged animals that do not have GSD1A. Again, they can have one copy that makes them a carrier for GSD1A. But then we see that in the liver, we're able to increase glucose 6-phosphatase, depending on whether given at a newborn, though it's lower, and they think some of that is just, it's really hard to dose newborn mice. I will say that with personal experience. But a better ability to improve glucose 6-phosphatase activity to a much closer range to what their normal controls are. But in the kidney, we did not see editing. That was anticipated based on this model. And so what I'd say is, again, this is a liver-focused disease. We know GSD1A has correlates in the kidney because that is another place that gene is very important, but this is not anticipated to change the genes of the kidney. I'll come back to that. But if you see these images, I'd say it's very helpful, right? So this is the liver. That brown represents editing, or sorry, I should say activity. And I mentioned those 2 units before, right? Those baseline animals who were not treated had a level of G6Pase activity of about 2 units. This is the range. Their lowest editing activity and their highest was 33 and 289. That is drastically different, and even 33 kind of has very significant benefits to activity within that organ. Yet when you compare it to the kidney, really, you don't see that brown coloration. That previously published data demonstrated that restoring greater than or equal to 5 units, okay, so we started at 2 and the range went from 33 to 289 in the treated animals, that is supportive of having a significant impact. And that I mentioned there was no significant impact in renal and kidney glucose 6-phosphatase activity They did actually see some improvement in renal measurements. Again, we don't understand everything about the kidney disease and glucose and GSD type 1A, but some of it is because it's an organ that stores glycogen and has similar issues because of the abnormal protein. But some of it is if your liver is churning out acid and has other problems with metabolism, that secondarily affects the kidneys, and that's largely why it's been demonstrated that improved metabolic control on the level of the liver can improve the kidney control. Okay, so did not see edits there. In a mouse model, they did, though, see some kidney improvements. Okay. These next couple slides are going to compress the rest of a long paper into much shorter bits where, one, right, when I talk to my patients, especially the parents of my patients, fasting is the first question, right? So these mice were able to survive multiple 24-hour fasting challenges through 1 year. That is a long part of that mouse's life. So this is anticipated to be a permanent treatment. Again, we do not have data in humans because it's changing your own DNA. So even as your cells multiply, it carries that original DNA. And so it's reassuring to see that in the healthy gray bars versus the GSD1A mice, 20 weeks after treatment, they are able to, so 0 hours, is at the start of a fasting study versus 24 hours is 24 hours into a fasting study. Mice with GSD1A are unable to fast more than 60 to 75 minutes. And that 20 weeks, 35 weeks, and 52 weeks at 24 hours, virtually all of the mice were able to tolerate fasting above 60. Okay? So this was an effect that was seen, that was maintained for a year. And again, it's harder to study because truly untreated GSD1A mice did not survive beyond 6 weeks. Okay. So normal serum metabolites, again, we talked about it in that initial mouse model, and again, that's what's being treated here, is that in the untreated group versus the Beam 301 group, and I mentioned they started treatment at, depending at newborn or 3 weeks, but this is after 3 weeks of treatment and after 1 year of treatment, again, looking to say, is this going to be a lasting treatment? You can see that that teal bar for treatment versus that gray bar for control really remains very similar, and that NS means non-statistically significantly different, that although there's differences in each group, overall, they're matching. We see that in cholesterol levels, triglyceride levels, lactate levels, and I will again highlight lactate is not drastically different in mouse models, as well as uric acid. Okay. Normal liver size. So I think I put it here. Yep. So normal liver size was maintained over a year after a single dose of Beam 301. So in this reference group, again, in this mouse that had the has the mutation of R83C, so has GSD1A, has a 3 times larger liver compared to the controlled mice. But when you look at 3 weeks, 8 weeks, 21 weeks, and 53 weeks after treatment, they are far more similar in size. I will say, highlighting that these are, again, I'm a very visual person, but you can see in this mouse with GSD1A that's untreated, these clear areas on this type of stain largely reproduce kind of where the glycogen is, versus you can see a far less amount of clear areas in the animal treated. And in the same way, it's red. It's hard to see, but you can see this hazy background where it's a little bit more lavender with red and blue, versus it returns to this really blue background without a lot of red. That red represents lipids stuck in the liver as well. I'm going to highlight. So one, we talked about these GSD1A mice, unfortunately do not live very long. So how do you see adenoma development if adenoma development takes a very long time? So, right, I would love to show you a picture and say these mice had less adenomas and less risk of tumors. There are other studies that have different ways of causing GSD1A in mice, and so they can, again, it's their ability to live that long to assess it, that They did not see any hepatic tumors in the treated mice at 53 weeks, so unfortunately there is not more mouse data to come on that. Normal body weight was maintained. Again, I showed you that those initial mice with the disease, again, a term that we often use in humans is failure to thrive. Anybody who's a family who has a type of GSD, many people often in infancy presented with that term failure to thrive, that really they were not growing normally, their weight was not normal. I will say they measured weight and growth of different organs in a lot of different ways, but truly to show you the weight data, that again, there was a big difference, 50% of the weight in the mice with GSD1A, and again, we saw this match, and that NS means that in the two groups, when you compared them statistically, there was not a significant difference. And then survival, right? I keep mentioning, unfortunately, that those untreated mice live an average of 6 to 7 weeks, versus wild type is another term for control. Again, that's a mouse that does not have GSD1A, versus that single dose of Beam 301 demonstrated that they can live a similar length of time. My mouth gets drier. Okay, how am I doing? Good. So, I am happy to answer questions on the end. That is what we know from mice. And as I said, this is an ongoing trial. Started enrollment approximately 1 year ago. I'm going to talk about some of the details of the trial and how it's designed, but this is a first-in-human trial that is referred to as a phase 1/2. Phase 1/2 trials really are built to study the safety and efficacy, so that means how well does it work, of Beam 301 in people with GSD1A and at least one R83C variant. Again, this does not address all of GSD1A. So part of it is collection of safety, but part of it is, right, they did this in mice. They tried different doses at different ages. And so what is the best dose in humans? We do not know yet. And so a study like this starts with multiple groups trialing different doses. I'm just hit people off at the pass, happy to discuss it. But one of the misconceptions is, oh, higher doses are better. We do not know that, right? So we don't know that. Higher doses may or may not. I did show you in the mice, right? Higher doses seem to have more editing. We don't yet know that in humans, and we also don't yet know higher doses kind of from a risk perspective. Again, before this came to humans, there was a lot more safety done in animals. Again, it is just a very different group. So I do not recommend having that assumption that higher is better. We do not know. That's the issue. So it was announced in March that we were able to move up to the second cohort, so that is a second dose exploration. And again, how this study is designed is that it was designed to take up to 36 people. This was approved by the FDA in at least 3 cohorts, so at least 3 planned doses, right? After every cohort, somebody asked me this, we have to reassess kind of what was the safety and how well did it work. And so that is how you decide to go up. Okay. So what are the key objectives of this study? Is that safety and efficacy, efficacy measured by time to hypoglycemia, starch supplementation dose, serum glucose, and other metabolic markers. Again, I'm going to highlight triglycerides and uric acid to try and confirm the optimal dose when used in humans. This is not an exhaustive list of key inclusion-exclusion criteria. Again, in many ways, when you do a study, I mentioned mice. Mice all match each other very genetically. We want a broader group, but we also want a very clean group, right? If we're measuring to see changes over time, if somebody starts with a more significant disease feature, it can be hard to kind of carve out, is this their underlying GSD1A, or is this an effect of the drug? And so that's the reason really for a lot of the exclusion. So some of this addresses adenomas, and I'll say specifically kind of adenomas over 5 centimeters are excluded from this study. If somebody has an adenoma in that moderate range between 3 to 5, that's growing more rapidly, that also is an exclusion. It's not on there, but triglycerides over 1,000, as well as liver function tests. So AST, ALT is looked at a different marker for health of the liver. Those also need to be within the normal range. At this time, any previous treatment with cell, gene, or viral vector-derived therapy is not possible. I will highlight it. Somebody having a lipid nanoparticle therapy or an RNA therapy can be included, and it's in part because it washes out so fast. RNA does not have a long life, and so that's the reason that they can be included versus somebody who had a viral vector or gene therapy. And that doesn't mean that if this is an approved drug in the future, somebody who had those, were involved in those studies or had those treatments, there's no intrinsic reason why they couldn't do a different therapy. It's just in a study. You need a very clean, understandable sample to make conclusions. Okay. This phase 1/2 study is open for enrollment within 3 sites, Connecticut, Texas, and California. I will highlight members from those teams. All 3 of those teams are here at this meeting at this time, and it is still enrolling individuals who are interested. You can email Beam Therapeutics if you're interested. Again, I think all 3 of the sites have websites up as well if you want to email them directly. And again, just emailing does not mean that you are enrolling. I'm giving you a nice overview, hopefully a nice overview. There's a lot more information to discuss when considering a trial, which again, I'm going to highlight this is a first-in-humans trial. So how is it designed? I tell most people it is very front-loaded, that already when you as a family are working on GSD, that is a lot of work in your life, that this is an addition, and truly the trial in all of the centers tries to minimize that as much, but we need data to demonstrate that this is a usable potential treatment. So there is screening where there's an in-person visit. There's this longer enrollment period, again, having very stable baseline cornstarch needs, dietary, blood sugars, right? We need, just like the mice, we need a good baseline. Humans are not mice, but we need a good baseline to compare to after treatment. You then have the infusion, which, as I mentioned, is through an IV into a blood vessel. This study is designed as only a single infusion, as is the data that I presented from mice. And then currently this study is approximately 2 years, that has 4 overnight hospital stays, and then a mixture of other appointments that can be in person for almost like your clinic visit, or some of them can be virtual by video. There was something else I wanted to say and I forgot about it. Oh, I'll mention that during the course of this is, again, right, we need to see what are people's cornstarch needs compared to the baseline, what are dietary changes, what are blood sugar. So throughout this, there's a fair bit of monitoring, which, as I said, it is already a lot of work to have GSD, and so I recognize that that is on top. Okay, this is a big conclusion slide, but nucleotide editing with an adenine base editor is a form of gene editing to address point mutations. Lipid nanoparticles can be used to carry genetic therapies. Again, I did not present data specific to the lipid nanoparticle in this study, but overall in other studies, it generally has a reassuring safety profile. Beam 301 targets only the most common mutation in GSD1A. And again, I do not have insight. I run the trial at one site. This does not mean I'm part of the company or anything else. Really, as somebody who treats all forms of GSD, I really will say that, right, my hope and dream, and that's on the next, that having substantial gains in one disease might help everybody. Preclinical mouse data demonstrates a high rate of specific editing with very low rates of making edits that are not desired. Improvement in glucose 6-phosphatase in the liver, but there's some variability. I didn't highlight that, right? But that 33 versus 283, those are 2 very different numbers. So even in mice, there's variability, and humans are far more complex. These mice, though, demonstrated that they were able to fast 24 hours versus 60 to 75 minutes. They saw metabolic markers improve and improvement in liver size and body weight. Truly, the kidney is not anticipated to be edited at all, and we do not know the benefits or risks to the kidney, and really, it's very hard to assess adenoma risk in mice. And they did demonstrate significantly improved survival. I briefly went through a phase 1/2 clinical trial. What it aims to do is safety and efficacy for people with GSD1A with at least one R83 variant. Okay. I started saying this. Again, my hope and dream is somebody who has the pleasure of working with people and families who have GSD1A and other GSDs, that really I'm very honored and excited to be any part of this, looking towards a future where I think there's a lot of hope. There's a lot of people to thank that I get to collaborate with on these. Again, there are people around the world that have really participated in a lot of this research. This happens to be individuals who are participating at centers that were participating in Ultragenics. And again, right now, BEAM is a small trial, but I get to look towards the future with hope. Again, I had the incredible pleasure of, in my younger days, working with Dr. Wolfsdorf and Dr. Weinstein at Boston Children's, and that I have the most incredible team. I feel lucky every day for those in my group at Connecticut Children's, University of Connecticut. But most of all, really thank you to all of the participants in any clinical trial. It really should take a lot of thought and support from the individuals who you're talking to. And really giving of your time and kind of your experiences is a tremendous contribution. Really thank you to all the patients and families for your advocacy, invitation, and I learn more from my patients and families every day, so thank you very much. Okay. I have a few minutes left. I'm happy to answer any questions. Turn the back.
On my way.
Thank you very much. That was an excellent presentation. You mentioned there was another accidental mutation that was caused. Was it an earlier identified mutation in GSD patients, or is it completely a new mutation?
So the two technical term is off-site edits. So the one edit, that's a great question. So I think you're asking specifically about that one edit within the glucose 6-phosphate, the G6PC gene. I actually don't know off the top of my head. That's a really great question. I'm not even sure if I fully have that answer. I will have to look back. So I'm happy to answer that question if I'm able to. Great question.
Thank you. Really
You can sometimes
appreciate
make prediction, but great question. I will say the other area, that other gene, that intron part of the other gene does not have anything to do with GSD.
Okay. Thank you very much.
Oh, thank you.
Need some waiting music.
A couple of questions. So I have two young grandchildren. Now, they don't have the R83C, they have a D38V. So how transferable is it once you figure out this, that you're able to then edit that? Is that an easy move, or is that a whole nother trial that's unknown?
Yeah. So I will answer it as somebody who is no longer in the lab. Easy for me as a doctor to be, as a medical clinical doctor who sees human patients now. Sure, I will say in reality, it will take a lot of work. Some of the basis for some other mutations has already started in mice. As of right now, I cannot tell you a timeline for human trials or anything else because they're not there. So I really would like to answer that question clearly. I don't know. I'd say these techniques are very new, but I'm gonna highlight a positive of them. So, what is it, 6 months, a year ago, a patient out of CHOP, I believe, with a different metabolic disease, and they were able to form a designer drug for one patient, right? So these are different, again, as somebody who spent more time in the lab a decade, 10, 15 years ago, which I personally don't think was that long ago. The ability to do these type of techniques fast is drastically different in the last 10 years, drastically, and that is CRISPR, that it would take a decade or more to potentially develop that in the past. And so I will say that does give me a lot of hope, because the ability to change this technology to affect in different ways is much faster. Still does not answer your question, but truly I have more hope now than I ever would have thought possible 10 years ago.
All right, that's helpful. And last question, the trial is on young adults, I believe. So assuming that all works, when would you move that to younger children, or is that, again, a whole nother trial?
Yeah, so generally these phase 1-2 trials, because they're focused on safety, focus on adults. So this current trial is greater than the age of greater than or equal to 18 years of age. That truly, even I think it was mentioned in the prior part, right, kind of having a child understand the risks and benefits of participating in trial is drastically different than having an over 18-year-old. So at this moment, it would be a different trial. I will say in rare diseases, especially now, sometimes things change, but for right now, over 18. I will flip to saying that these type of technologies, because there are other metabolic diseases that are not survivable past infancy, and this is part of the reason that they tested it in newborn mice, is that, right, again, that I get to stand forward and kind of dream of a future that really these are technologies that do have possibilities in infants, children, et cetera, but we're not there yet in humans.
That was fantastic. I'm going to corner you after, and I'll share with Priya too. I have one question. So I know you touched on when patients are, or mice and potentially patients are treated, that of course you can have an inherent just immune or allergic response. But could you touch on, I know a big concern about AAV gene therapy is, right, we're not quite there at being able to redose because you mount an immune response to the AAV. So could you expand on that a bit? And with that, if I understood correctly, the editing efficiency was about 50% in mice in the liver. I don't want to quote if it was
The
higher.
activity efficiency?
Yeah, yes, or at
Correct.
least
It was close to 60%, correct.
So I'm just, more, that's more, right, what a carrier would be. But just if you could expand on the potential, if any, of, again, I'm not quoting you, I'm holding you to like potential redosing.
Yeah,
and what that would look
I'm smiling. I didn't ask you to be a plant, but those are two beautiful questions. Okay, so the first one, AAV8 versus lipid nanoparticles. And again, lipid nanoparticles is a new technology where most of the information we have is right under the skin for vaccines. There are other trials, there are other things that use them through the vein, but that's much newer. So the difference is, right, our bodies are designed to make antibodies against viruses, right? That's what keeps us all alive. And so although that is a beautiful mechanism, if I need to carry a drug into your body using even kind of a defanged virus, kind of a virus that doesn't have the same risks as other ones, your body is going to respond to antibodies because that's what it's supposed to do. You can respond to different antibodies, so I can't tell you that there is zero risk for another agent like a lipid nanoparticle, but that the outside is actually just like the lipids, largely just like the lipids you have in your body is not going to respond in the same way. Alternatively, I will say, right, viruses go into your body and some have very severe inflammatory reactions and some don't. Lipids can be very irritating, other biological materials, so again, meaning things that are already in your body. If I give you a low dose of them, you're also at risk for other inflammation. So thank you for that they are differences. And there were other talks, unfortunately one that I missed, that one of the big difficulties with adenoviral technology, I mean, one of the benefits is it's been around, AAV technology has been around for a long time, so it's nice to use something that you know. On the other hand, overcoming that antibody response to be able to redose it is a problem, not just for studies focused on GSD, but for the field as a whole that's trying to be addressed with other medications. Again, doesn't mean that it's impossible to have somewhat similar issues, but that same kind of antibody response is not expected to be widespread. Your other question was great as well, the 50, 60%, and used an example that I often like to use. So I will tell you, in my dreams, in a human, right, so people with the most severe mutations, R83 being one of them, really kind of that enzymatic activity is close to zero. If you inherited one or both genes from your two parents, each of your parents has one copy, most commonly. And even though some parents can be a little symptomatic, they are not dependent on cornstarch to sleep through the night. They're not dependent on multiple doses. So that's about kind of having 50%. Okay, it doesn't work perfectly like that, but just to give you an example that, and again, I will say again, it does not work perfectly like that in genetics, but it's an example that really having a significant edit can make it, doesn't get rid of GSD. Okay, these treatments do not cure GSD. It is a different GSD. It's a different disease. And so that I will say the mice got up to 60% correction. Again, this is I love being wrong when I say things like this, and this is my opinion, not the company's opinion. I still am like, how do you correct 60% of a human liver? Like, that sounds above and beyond my wildest dreams. Truly correcting 10%, 20%, it might not address everything, but truly from a fasting perspective, from kind of a dose need for cornstarch, still could have significant benefits. But you are right, they were able to demonstrate in the mice 60%.
And great talk, thank you. So in tune with that very good question, it looks like the trial currently is planned to do one dose. And is that dose 0.1? So the safest lower dose. And I'm assuming there may be pre-medication prior to that infusion to mitigate some of the potential immune challenges that you can get with the lipid nanoparticle. But in connection with the previous question about the CPS1 example at SHOP, they did a second infusion at 0.3, which based on the data shown here in the animal side, seems to still be a little bit more desirable in some other data. So I wonder what was that decision making on starting at 0.1, probably just a safety kind of issue, and if there are plans for the further phases of the study to do higher dosing.
Yeah. So the human dose is selected. I don't even know if I'm not sure if I can fully answer that question, but I'll say the human dose is selected correct, is starting kind of in the range of that low dose in the mouse model. That even with the low dose in the mouse model, we did see metabolic benefits. We did not see that level of correction being 60%. I believe it was somewhere more like 20 to 30% in that low dose. So it is not just, I was explaining this to somebody else, generally in kind of very big trials, if you're doing a big trial of diabetes, a lot of that phase 1, that safety is giving tiny doses to see risks. In a rare disease population, you can't do that. So it is a dose that is similar to that mouse dose that had some efficacy to assess the risk, but was not at that 60% correction. The second question is an excellent one. So the question was that kind of other studies sometimes will use a second dose. Again, I mentioned this is in mice, demonstrating that it likely is very long-lived, if not the duration of somebody's life based on the science. And again, I use words carefully because humans do not always read that textbook. That theoretically, if it's a permanent edit, is that will last your lifetime. So theoretically, we think maybe. Don't you love all those maybes? On the flip side, right, that we're doing a dose escalation trial. So, and I mentioned a lot of variability in mice. So is there a potential benefit in the future for doing a second dose? I'd say based on the science, yes. Based on the current design of the trial, not at this time. I would say based on the science, again, when we talk about antibodies and redosing AAVs, there are other obstacles that we don't anticipate. I will say even the Moderna trial, although did not kind of widely publish at the end, the earlier data that they demonstrated did have some data, I think, of multiple doses with a lipid nanoparticle. And there are other studies that have done multiple doses. Sounds like that one as well with a lipid nanoparticle. So possible. I don't have yet an answer in humans studies. I think Dr. Krishnani has a question.
Hi,
Yeah.
excellent, and what a nice way forward, right, for the field. But two questions. How would you really measure the amount of correction in a clinical trial of patients? I understand in mice that you can go in and do it differently. So is it more by metabolic correction? And maybe you'll have it initially and then over time there might be a waning effect. So that's one question.
Mm-hmm.
And the second is really safety and off-target. What are the precautions or ways of monitoring for off-target impact with this kind of work?
Yeah. So two excellent questions. Let me do the second one, then I may need a reminder for the first. So I will acknowledge, right, like this is, I'm open that there is likely something more behind that. Well, actually, let me answer the first question first. So pointing out that really kind of finding what's called a metabolic marker in GSD1A is not easy. So I mentioned those people with GSD1A, probably if you're older than 20 or at least older than 30, you had a liver biopsy at some point in your life. People younger than that, it's a bit more of a mixture. And the kids born today are not routinely necessarily getting liver biopsies. So, right, the best way, and in the mice, is that they actually look at a piece of the liver to see what that activity level. This study does not have that as part of it. And so that would be the cleanest way to do research on this. There are a few newer studies, and I'm looking for Dr. Dirk, so there are a few newer studies looking at other potential markers. As of right now, this study isn't designed to do that. We are using secondary markers, which other studies have used, which are imperfect. And so the secondary markers, when I mean that, it means like we're not actually looking at the liver and knowing like this enzyme is working. We're looking to see, can somebody fast longer? Can somebody take less cornstarch? And those have their own layers of having difficulty interpreting. So I fully acknowledge that we are not directly looking at it. The second question for off-target, I'm definitely interested. I actually can think of like, okay, there might be some ways that I can think. As of right now, it is collecting generalized health data, that there is no disease process known kind of from human cell data and honestly database modeling efforts, that there's not an identifiable phenotype, there's not identifiable features for that offsite target. And so other than general health and kind of the massive monitoring that's done in a clinical trial, there is no specific right now. I see a question all the way in the back. Yeah.
Thanks again, really great presentation, presenting like complicated data in an understandable way. So trying to bridge from, again, the mouse experiments to the clinical trial, and it seems like this was really dramatic in terms of the correction for the mice. And what would be, you know, a true success, really life-changing as an outcome in a clinical trial for those participants? So based on, you've really got some keen insight to the whole process here. And what would be a huge success for you?
Yeah. So I like that last part of how you phrased that question, because, right, success can be defined in different ways. And there was a study, looking to see confirmation, I think that was published, kind of asking, right, the first thing is, what do you, if you have GSD1A, what do you consider a success? So that really is a measure that needs to be done and is being done, right? Because I think all of us measure success in different ways. I kind of commented, again, this is not the study, and then I'm happy to, right? I honestly, when I talk to patients and their families, parents have very different views of success than an adult patient. Parents' view of success is fasting through the night. Adult patients would like to do that as well, but adult patients, some of them will be like, no, even more important to me is to kind of be able to expand my diet and my ability to eat calories outside of cornstarch. So, right, so one, defining it as somewhat there. Again, I think it's hard, I'd say as a doctor, right? I would love as much correction as possible with safety features. That's the easiest answer. The second answer is I try and put myself in other people's shoes, and I apologize if that, right? For me, again, as a parent, I really want somebody to have safety to sleep through the night. And again, other GSDs have different needs, and so it's not the same, and I'm not kind of comparing, but even being able to cut down the frequency of doses, as I said, 60% is a level where truly, theoretically, somebody could be without cornstarch, right? The mice demonstrated fasting for 24 hours, not needing added glucose support, having their liver get closer to normal in size in normal labs. Again, I'm a realist, and I think humans are complex, and I think achieving that goal would be mind-blowing to me. I still have a hard time wrapping my head around it. So for me, really having significant safety improvement overnight, and that by definition, my hope would be to be able to decrease cornstarch doses without added risks. Very long-worded answer to say there's lots of them, and I don't know. Dr. Kishnani, and I can repeat if I... So to highlight that question, so one, again, that this is a potential therapy that is targeted to the liver and is not addressing the kidney. One, there's another way to think about it, that individuals who have had liver transplants with GSD1A, they do have some risks to the kidney. It's a little bit hard to sort it out because liver transplants have drugs that can have danger to the kidney. But so the question comes back to, are there any ways to kind of think about it in a different way? And are there any potential risks to the kidney with treatment of the liver? And the answer is, we don't have those answers. And so that's, I think, a great highlight of these trials that I'll comment, this trial and others. This trial, it's 2 years, but the anticipation is for people we will encourage, research is always voluntary, but to sign up for an additional 10 years of follow-up to answer that question. Again, in the mice, they saw some improvement in the laboratory markers for kidney health, but right? Mice only live a year or two. I want to know, as best as I can, I would love to tell somebody, oh, in 30 years or 40 years from now, like, these are your risks to your kidneys. These are unfortunately not. We're not going to have answers to all of this. During this study, we are collecting information on the size of the kidneys, protein that comes through the kidneys, something called albuminuria or proteinuria, kind of other measures of kidney health, but we won't have those long-term answers.
Unfortunately, I have to cut you off there because
Nah.
it is officially lunchtime, but how about a nice big hand for our speaker? Thank you very much for your time.
Thank you.